All right, while we're waiting for folks to come in, if you could pull up your um, macromolecule chart, we're going to look at that today. All right, live stream is up. All right, so good afternoon. While we're waiting for folks to come in, if you would get your macromolecule chart out, we're going to look at lipids today. So get your chart out. And then go to Canvas. We're going to see if we can't get through lipids and nucleic acids today. So again, while we're waiting for everybody to come in, get your macromolecule chart out. We're going to give it a quick review, and then we'll move on to lipids. Okay, why is my chart redirecting me? Let's see if I don't know what that is. So we will try it again. All right, so good afternoon. If you just joined, we're on camera. And we are looking at, so right now you should have, and again, my chart doesn't have everything on it. Yours should be full of information and any tidbits that you can find to add to it. Make mine a little bit bigger. There we go. Oh, and I know this is the group that gets a little flustered about this chart. So everybody should have the same pictures on the chart. I know, like I said, my chart doesn't have everything that you need to know on there, but you need to have filled in these first two columns with information from either the videos we've watched or from Canvas. You don't need to submit Nearpod. The Nearpod records the information, so unless it did not let you finish, you're good. You don't have to, Nearpod assignments don't need to be submitted to me. I take the grade from Nearpod and I put it in grade in Canvas. So if you were trying to submit something to me, as far as the Nearpod was concerned, you can't, because it's not an assignment that had a Dropbox to it. I recorded the information off of Nearpod. They keep a grade book. And let's see, Dallas. All right, I got your scores, but I don't know whether you wanted to go back in and finish them, because some of you look like you didn't finish the Nearpods, but didn't let you go to the next video. And there was no close date on that. So I would try again. With the Nearpods, I don't have let me check before I say that, and it's not true. Let me check. Let me check. All right, because we're going to do Nearpod again on but there is no Dropbox for the Nearpods. Some of y'all have 0, or you didn't do anything, and some of y'all have 1. Kudos to those of you who completed all of the Nearpotics activities. So let me look at my reports. Actually, I'm going to stop sharing for a moment, because it does give scores and names. Okay, stop sharing. Okay. Like I can't click on too many things at the same time. It goes, what are you doing? All right, so it was launched on the 21st, but there is no closing date on it. So as long as you have the code, you should be able, if you run into a problem, it might be that you need to restart your computer. Yes, Dallas, I see that you were in, but you weren't in. So restart your computer. There's no lockout thing on Nearpod activity. So as long as you have the code, and you all do, because it's part of the assignment. So you can get the code from the assignment tab, but you can't add a score to it. Okay, I have to take the codes from the grades from Nearpod and put them in the grade book. So let me see while I'm looking at this. So Dallas, you need to do yours. Jayla, Marshall, and Desiana. Kinsley, you all need to do carbohydrates. And Neve, I think you were absent. You need to do that as well, because you have no score because you were absent. So Neve, you have both proteins and carbohydrates to do. And again, if you look at the assignment tab, you can get the code, the Nearpod code, and you can do it from there. Isabella, she here? She also was absent. Not here. All right, no, she's not well. Um, but yeah, with the Nearpod assignments, as long as you have the code from the assignment, you can go in and complete those and finish them. But you can't add the grade to the grade book. There is no drop area for that to be done. Okay. All righty. That being said, because there's a couple of you. Ethan, you need to do yours too. So I got Ethan, Isabella, Dallas, Kinsley. Um, that's it. Troy, you need to do proteins. All right. So if I called your name, you need to make sure that you go in and do those. But not today. Not today. We're moving on today. So please focus on what we're doing today. All right. Back to sharing. So let me share again. All right. So again, we're looking at your macromolecule chart. And again, I want to say that my chart doesn't have what your chart has on it, because you're supposed to go and add information to the chart. The more information you add to it, the more help it will be to you. All right, remember, this chart is for you. This is a kind of a cheat sheet. So you want to make sure it has as much information on it as possible. All right. So everybody should. All right, so what we're going to look at today are the lipids. And if I need to resend everybody the pictures for lipids, I will do that. But hopefully you already have these All right, and my Zoom disappeared. All right, it's yes, I don't know what happened. It's one of those briefly where Zoom is not responding. And I don't have time to send messages. Okay, it's responding now, and y'all are back, and we are sharing. Okay, so again, in review, we are looking at macromolecules. These are molecules that are, like it says, big. They are part of living things. They are not in themselves a living thing, but they are essential to life. All living things are made up of these um, 5 macromolecules. So this is why we're taking the time to look at them. So the first one we looked at was proteins, and proteins, as far as elements are concerned, are made up of carbon, hydrogen, oxygen, and nitrogen. You're going to find that all of them are going to have carbon in it because that is a characteristic of living things also. All living things are made up of some type of carbon-based molecule. If you watch sci-fi stuff, sometimes they refer to human beings as a carbon-based entity. But everything that we're looking at is going to have CHO for sure. Now, proteins have nitrogen. That kind of makes them different from the other ones. If we looked at carbohydrates, they only have carbon, hydrogen, and oxygen. So they don't have they don't have nitrogen as part of their general makeup. All right, so now we're going to look at lipids. And lipids are going to be the same thing, and I'm going to type that in mine. Actually, let's go. All right, before I type that in mine, let me see over here. All right, so have your Canvas open. Have your macromolecule chart handy, but also have Canvas open because we're going to take the information off of Canvas first. All righty. So everybody should be in Canvas looking at lipids. And did you disappear? All right, here we go. All right, so like it says, you may not have heard the term lipids, but because we are science students, we're going to call them lipids and not fats, but they are fats. So that when you go to your kitchen cabinet and you take out that Crisco or whatever oils you use, they are lipids or fat substances. And there are different kinds. There are solid fats, liquid fats. So under here we have examples. So looking at this right here in your last column on your chart, let's start by putting these in. Okay. And let's just put that part right there. Let me copy. See if I can't do that. Want to copy it? I'm going to go to my chart. Come on, chart. And I'm going to over here in the column of examples. I am going to paste the examples. There we go. So that's what everybody should do. Examples of lipids include fats, waxes, oils, and steroids. And back that part up. Okay? That's what you do. Now I'm not going to add everything to my chart but I'm going to tell you what to add to yours. But that one I decided I'd help you out and do that one. All right. So you don't need to copy me. You just need to know where. So I got it from right here. I got that from Canvas. Now, all right. One of the things that makes them interesting, and I'm going to copy that one, help you with that too. All right. So you have a picture in the box already. So underneath the picture, I'm going to move mine up a little bit. So underneath the picture, I'm going to put what they do. All right. They are so something in so this is our column that says distinguishing features. One of the distinguishing features of a lipid is they are insoluble. That means they do not dissolve in water. So you know this, you get grease and it sits and floats on top of the water. Okay. Um, and some of them are liquid, again, at room temperature, but what they do have is high amounts of energy. Now, when we looked at the function of these, we looked at function. So you should have as function underneath your protein, that proteins are made for structural, they make up the tissues of your body. Carbohydrates are a good source of energy and structure for them too. Lipids are also going to be good for energy. So everybody can give you energy, but the main function of a carbohydrate is to give you energy. That's all it really is important for. So when we want to get some more energy, we eat some carbs. Um, you've heard people talk about weightlifters and people who exercise, they're carb loading. They're carb loading because they're trying to get energy. All right. Lipids, on the other hand, are something that we store. And I think up here, we said we store um, starch as glycogen. So we don't exactly go to lipids first because they're big molecules, they're hard to get energy from, but because they are huge, they have lots more energy than a carbohydrate. Okay, so we're going to come back to that in a second. So that you should put that under there, that they are insoluble in water. You should have anybody need this picture of the structure because we were looking at the fact that it as a unit, the fatty acid has an end that is hydrophilic, which means that end likes water, hydrophobic and doesn't like water. So this is when you have this, when we look later on, when we look at the cell membrane, the hydrophilic part will let water in, the hydrophobic part repels the water. You need a photo. Okay. I'm gonna have to, every time I try to copy and paste it from here, it doesn't let me. Hold on, let me try this. Copy. All right, Dallas, let's see. Yay, it worked. Well, it liked that picture. All right, well, I put that. All right, let me put it for everybody since it worked. Maybe that's what I have to do. I have to always just to control V. All right. So for those of you who didn't ask, there's the picture in the chat. So if you don't have that picture in your box, there you go. All right. So they have a special little function. All right. So we'll go back to Canvas and let's look at what else we can find out about lipids. All right. So you should have opened your box. This was one of those boxes that you need to open up. And again, make sure you open it because this is where you're going to get your stuff for you. All right, so this part, and can I copy? I can't copy and paste this part, but you need to put this first sentence in your first column. Okay, so like I have for the other ones, this is made up of carbon and hydrogen. Okay, a lot of carbon and hydrogen. It's got a little oxygen in it, but it's mostly carbon and hydrogen and sometimes phosphorus. So you need to type that in your first box. Okay. I'm gonna stay right here. Wait for you to do that. We already have the example, so don't worry about that part. You're writing lipids are macromolecules composed mostly of carbon and hydrogen, and you can do the initials like I did, and you can abbreviate like I did. Let me come back over here. I keep saying I'm not going to type it. I keep doing it. All right, so you can abbreviate like I did. Made C. All right, you got to remember that P is for phosphorus. Oops, I spelled that too wrong. Spelling right would be helpful. All right, so you put that on your box. And you can shorten it just like I did. Or you can write it out long. All right, so I'm going to hit my arrow and go to the next one. Characteristics of all lipids. Characteristics means that's going in your box as well. So we just wrote that they're insoluble. We already wrote this one. They store high amounts of energy. So we already wrote that. This is what you need in that first box. They act as waterproofing. So the surface of leaves and plants, they act as waterproofing. The reason why you don't get soggy when the water gets on your skin, because you got a little waterproofing on the surface of your cells. They are chemical messengers. When they are steroids, they are chemical messengers. We already wrote that they store energy and they form the membranes in our phospholipids. So put this under the first column. We already have the first box in our second column. Add importance of lipids in under the function. Under where we wrote carbon and hydrogen, write this. You're putting this on your chart. All of this is going, you're not making a separate notes paper. You're putting this on your chart. If I could copy and paste it, I would. But you're going to put everything in this second box is going to go right here in this first box. This is the reason why I told you to do this in Word, so that as you enter things, the box will get bigger. Okay, the box should expand as you type more information in it. So you shouldn't have to write your information under the chart. This is where I was trying to make an extra. Oh, I know what I have to do. Delete, delete table. There we go. Now, okay, that's gone. All right. So you add that in this first box over here. If you need more room, you can stretch it out into, in fact, you should be able to move that over like that. And that gives you more room. See, all of these lines should be able to move so it'll accommodate whatever information you want to put in that box. Okay. All right, so all of that goes up underneath, made of carbon and hydrogen with some oxygen and phosphorus. I'm going to see what that looks like in a minute. Right, again, you're taking that information from the importance of lipids and we're putting that under our function column. Everybody should be going back and forth between Canvas and your macromolecule chart. The more information you put on the chart, when you take the test, you don't have to go flipping back all over pages. You have one document to look at. All right, this next one. So when we looked at, I'm gonna come in, you will too. When we looked at the structure of a monomer is made up of something called a fatty acid molecule. Okay, and if we blew this up, it was mostly carbon, hydrogen, and it only has oxygen up here on the end, what they call the carboxyl group, and then long chains of hydrocarbons, meaning hydrogen. So if you look at this picture in the polymer box, make this one a little bigger. And let me know if you need any of these pictures as well. In fact, let me just put these in. All right, but if you look at the picture of the polymer, you'll see that the ones in the gray areas are simply long chains of carbon and hydrogen all connected to each other. So this is why we say it's mostly made up of carbon and hydrogen. On the end, at the top, it's got some, let me see if this is going to work. Nope, that one's not going to copy. I'll try it again. Might have to go on Google to get another copy of this. I don't know why the first one worked and this one won't. Nope, it won't. All right, so while you're looking at that, let me see if I can't get this picture for you. Again, if I spell things correctly, I'll get what I'm looking for. Not the one I want. That's the one I want. Okay. And then all right, so note this is called a triglyceride structure, and that's the one I have on my chart. So that's the one I want to give you. Okay, there we go. All right, so for those of you who don't have the pictures on your chart, you should have them out. All right. All right, so again, you don't have to write anything from Canvas about this because you just need to look at the picture for both of these molecules. So our monomer is a fatty acid molecule. All right. And there are three of them. So this is why it's called a triglyceride. So you might need to type that word up underneath it. Let's do that. Type the word triglyceride. Copy and ah. There's a thing that says undo. Anyway, let me do this. My picture disappeared. All right, that's a triglyceride. I'm going to put my picture back. It's Monday. All right, picture's back. All right, it's called a triglyceride because it's got 3 rows of fatty acids and they are connected. You see this thing says a glycerol backbone. So it has this backbone piece that connects 3 of these molecules that we have in the first box to each other. And that is a polymer. And you see how big that is compared to what we have up in the other ones? These are long, 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 long chains. Okay, which means they all, and later on we'll talk about the fact that all those little bonds, all those little black lines, we call those bonds, and it takes a lot of energy to break them. So when you digest things trying to get some energy, you get more energy when you break those bonds than you do with the carbohydrate. Okay. So the carbohydrates, you get good energy out of a carbohydrate, and that's the main function. That's all carbohydrates really are functional for, is to give you some energy. Lipids have, other activities, but they also provide you with a lot of energy. So this one over here, we said high amounts of energy because they store a lot of energy. When you digest them, they give you a lot of energy. Now, again, they are there to give you to insulate your body and give you warmth. And so the number one thing is not to try to break those down unless you're on a diet and then you start starving yourself of carbohydrates and the body says, I need energy. I'm going to find it somewhere. I'm going to start breaking down the lipid molecules in the body so I can get some energy. But it will start with the carbohydrates. It'll actually start with the carbohydrates, then it'll do the lipids, and then it'll go to proteins if it still needs some energy and you're not giving it any. Okay? All right. So everybody with me? Everybody got those boxes filled in? Like I said, you have more in your information box than I do. I'm being all right. So coming back here, triglyceride. So that's what we just looked at it. And tri meaning 3. That's the glyceride molecule. Used for high energy storage. You find them in vegetable oils, such as peanut oils and olive oils, and animal fats, such as butter. All right, they have lots of energy. They have lots of carbohydrogen bonds. Like I just said, when they break down, when those bonds break, they give you a lot of energy. All right, I said that very fast. All right, you hear this on the television a lot. This is not going on your chart. Maybe you can write this under your chart. You need to know the difference between and saturated fats and fatty acids. That's when they do your cholesterol and they tell you watch your diet. It matters whether you are taking in good fat or bad fat. Okay? So saturated fatty acids contain a maximum number of hydrogens, and they're in animal fat and butter. Unsaturated fatty acids contain more double bonds, and the double bond causes a chain to kink, meaning it folds on itself. So we pack together. So guess what? The unsaturated ones are the ones that are not good for you because of that kinking effect makes it hard to break them down. So your body stores them. And they're the things that clog up your arteries. So you don't want those. Okay, so this is something that you can just put as a note down at the bottom or on the back or something like that, but you don't have to have it in your chart. All right. We mentioned them as examples, that steroids and waxes are examples. Steroids are signaling molecules. So you need to know the vocabulary, not necessarily part of the chart, but you need to know what a steroid is. And cholesterol is a type of steroid. So that's what I was just talking about, the cholesterol that plays an important role in the cell membrane. So we need cholesterol, we just don't need too much of it. All right. And they keep, though we're told to keep our cholesterol levels in check, the body still requires it. Steroids are typical flat molecules composed of rings. Waxes, on the other hand, are important in animals and other organisms such as plants. The waterproof surface, the surface of feathers, so they can be out in the rain and they don't get soggy. and they are based on 2 layers of fatty acids linked together. So again, you need to know what is a steroid and what is a wax, and you need to know that they are types of lipids. All righty. That being said, let's watch this little video. While watching the
Welcome
video, listen, but if you need to catch up on your writing, you do that too.
to biomolecules, the lipids. Lipids are organic molecules that are insoluble in water. In biology, lipids' main functions include storing energy, signaling, and acting as structural components for cell membranes. Let's examine 3 types of lipids. Neutral fats, acting as structural components for cell membranes.
All right, so make sure that this is something we have. So we talked about storing energy. We talked about the fact that steroids are signaling, and we talked about structural components. So you don't need to add that unless you don't have it.
Let's examine 3 types of lipids: neutral fats, phospholipids, and cholesterol. Neutral fats, also called triglycerides, are 3 fatty acid chains attached to a single glycerol molecule by dehydration synthesis. The fatty acid chains are the building blocks. While these chains can differ in length, each one has an even number of carbon molecules. Neutral fat stores energy fuel, insulates body tissues, and cushions and protects organs. Saturated fatty acids have single bonds between the carbon atoms. They originate from animal sources and are solid at room temperature. Other fatty acids don't bind their maximum number of hydrogens due to double bonding between carbon atoms in the chain. Fatty acids with 1 double bond are monounsaturated, while those with 2 or more double bonds are polyunsaturated fatty acids. Unsaturated fats originate from plants and are liquid at room temperature. Phospholipids are modified triglycerides, where a phosphate group replaces one of the fatty acid chains. Phospholipids, or phosphoglycerides, have a nonpolar fatty acid chain portion and a phosphate polar portion. 2 layers of phospholipids are the chief component of all cell membranes. In a cell membrane, the 2 layers self-assemble so that their water-soluble heads, or hydrophilic heads, form the surface and interior of the membrane, and the water-insoluble tails, or hydrophobic tails, face each other. Cholesterol is an organic molecule known as a sterol, or modified steroid. It helps build and maintain membranes and composes over 30% of all animal cell membranes. The cholesterol molecule has 4 interconnected carbon rings. Cholesterol helps to stabilize all animal cell membranes. The body uses it to synthesize steroid hormones, including sex hormones, and those of the adrenal cortex. It's also used during vitamin D and bile synthesis. Congratulations! You've completed biomolecules, the lipids.
All right, let's look at what Mr. Talks Too Fast. Let me see if I can. Because this is a clip, I'm not able to slow him down. Oh, yes, I can. Because he talks way too fast.
Our own use. Hard for me. Now we as human beings can't grow fruits or tubers, so we have to store our energy in a couple of different ways. The way that we tend to store carbohydrate energy is in glycogen, which is very similar to amylose or starch.
All right, let me preface this, but if you'll notice there was one big video and they've chunked it into sections. So he's kind of overlapping carbohydrates before he starts on lipids.
More branches and is more complicated. It's basically made up of the glucose that we have left over after we eat, and it sits in our muscles really ready to use, and it's also stored in our livers. It's generally a pretty short-term store. If we don't eat for like a day, pretty much all of our glycogen gets depleted. But over the longer term, the way that we store our energy is through Fat. All of our mom's worst enemies, the fat, which turn out to be actually really important and are the most familiar sort of a very important biological molecule, the lipid. Lipids are smaller and simpler than complex carbohydrates, and they're grouped together because they share an inability to dissolve in water. This is because their chemical bonds are mostly nonpolar. And since water, as we learned in the previous episode, despises nonpolar molecules, the two do not mix. It's like oil and water. In fact, it's exactly like oil and water. And if you've ever read a nutrition label or seen this thing called the television, you're probably pretty conversant in the way that we classify fats. But then, 99% of us have no idea what those classifications actually mean. Fats are made up mainly of two chemical ingredients, glycerol, which is a kind of alcohol, and fatty acids, which are long carbon-hydrogen chains that end in a carboxyl group. When you get three fatty acid molecules together and connect them to a glycerol, that's a triglyceride. These feature prominently in things like butter and peanut butter and oils and the white parts of meat. These triglycerides can either be saturated or unsaturated, and I know that when we put the word fat and saturated into the same sentence, it sounds like an evening at KFC. But here we're talking about being saturated with hydrogen. As you hopefully remember from our first lesson, carbon is very nimble in how it uses its 4 electrons. It can form single or double or even sometimes triple bonds. This means that if the carbon atoms in a fatty acid are connected to each other with single bonds, all of the carbon atoms end up connected to at least 2 hydrogen atoms, and one of them picks up a third. So the fatty acid is saturated with hydrogen. But when some of the carbon atoms are connected to each other with double bonds, all of those carbon's electrons are spoken for, and so they're not able to pick up those hydrogen atoms. This means that they're not saturated with hydrogen, and they are unsaturated fatty acids. To demonstrate, may I direct your attention to this jar of peanut butter. Here you can kind of see both kinds of fat. The liquid stuff you see at the top here, that is the unsaturated fat, which we generally think of as oils. The pasty stuff down here also contains lots of unsaturated fat, but also contains saturated fat, which doesn't have any double bonds, so it can pack more tightly and form solids at room temperature. And there are also other fat classifications that you've heard of. Trans fats, which everyone tells you never to eat. They're right, don't eat them. They don't exist in nature and are basically unsaturated fatty acids that instead of kinking, go straight across, and so they're extra super bad for you. Don't eat them. Omega-3 fats are fatty acids that are unsaturated at the 3 position, which is like right there. Then that's the only difference. But the reason why these are important is because we can't synthesize them ourselves. They're essential fatty acids, meaning that we need to eat them in order to get them. All this is starting to make me pretty hungry. But before we get to more food stuff, there are some unappetizing sort of lipids that we also need to talk about. So remember that triglycerides are 3 fatty acids connected to a glycerol. Swap one of those fatty acids out for a phosphate group and you have a phospholipid. And these make up cell membrane walls. Since that phosphate group gives that end a polarity, it's attracted to water. And the other end is nonpolar and it avoids water. So if you were to scatter a bunch of phospholipids into some water, they would automatically arrange themselves like this, with the hydrophobic ends facing each other and the hydrophilic ends sticking out to face the water. Every cell in your body uses this natural structure to form its cell wall in order to keep the bad stuff out and the good stuff in. Another class of lipids is the steroids. Steroids have a backbone of 4 interconnected carbon rings, which can be used to form hundreds of variations. The most fundamental of them is cholesterol, which binds with phospholipids to help form cell walls. But these can also be activated to turn into different lipid hormones. And so now we approach the most complicated.
All right, so like I said, he's segmented so that That same video will be continued later on. All right, so in the time that's left, I want, okay, and then read it before you go. All right, if you finish that, what we're going to do next class, we're going to look at nucleic acids, which is the next box on your chart. And we're going to do this briefly because later on we're going to talk about protein synthesis. So right now we're just interested in what is a nucleic acid in terms of it being a macromolecule. All right. So when you're finished, and again with that, I put these pictures on your chart while you are working on your lipid matching. I will make sure that you have the other pictures in. So everybody should be, this is where everybody should be right now. Doing your lipid challenge. Um, if you need time to work on your Nearpod, you got about 10 minutes left of class. So this is the time you work on catching up on what you need to catch up on. And again, if you are caught up, I'm going to make sure that you have the pictures for nucleic acids. Put those in the chat right now. But you should already have them. But just in case you don't, put those. Please work. Yay. All right. So a nucleotide is a monomer. And this second picture goes in our, well, too good to be true. Second picture won't copy for me. Yes, it did. All right, here's the second picture. All right. While you do that, let me make sure I did attendance. All right, so everybody should be working on the matching or fixing. All right, Akari, I don't know where you are, but you're not with us. I'm looking at the ceiling. I got 3 people over here. All right, after today, the Nearpods are late, except for Dallas. Nearpods will be late. So if you haven't done your Nearpod, Make sure you, and Neve, who was absent, but Neve, make sure you catch up on that as well. All right, anybody need more clarity on your chart? What should be on it, how to make it? Your first column should always have functions. What is that molecule mainly used for? Where are we going to find it? Mm. Right, and I am taking note of those who are logged in and not doing. You're doing something else in somebody else's nearby, doing somebody else's work. And then when you fall behind and need extra time and all those good graces that people are looking for when they're failing, and progress reports come out next week, I keep that in mind. The well of kindness does not extend to those who are deliberately not doing what they're supposed to do. All right, if you have no questions for me, I'll see you on Wednesday.